Imagine a patient beats a highly aggressive cancer.
Right.
Their doctor looks at the tissue biopsy and says, you survived because you had this massive army of immune cells. But what if that doctor was entirely wrong?
Which happens a lot, actually.
Exactly. What if the victory had almost nothing to do with simply having those cells, but strictly depended on who those immune cells were actively talking to?
It completely reframes how we view human biology. I mean, we are finally moving away from looking at isolated cells, and we're actually looking at the conversations happening between them.
Right. And historically, medical diagnostics just haven't been great at capturing those conversations.
No, not at all.
For decades, looking at a traditional cancer biopsy was, well, it was like taking a single, blurry Polaroid photograph of a massive crowd of people and trying to guess the mood of the entire city from that. It's just a flat two-dimensional slice. But now, we have spatial omics.
Which changes everything. If a traditional biopsy is that blurry Polaroid, spatial omics is, it's like having a Google Earth view of the tissue.
Oh, I like that.
Yeah, you don't just see a cluster of anonymous cells. You know exactly who lives where, what molecular signatures they carry, and critically, how they're physically interacting with their neighbors in the microenvironment.
But having a Google Earth view of a tumor introduces a massive problem, right? I mean, if I'm suddenly looking at millions of individual cells and the thousands of molecules inside each one and mapping all of their neighbors, I am going to drown in data.
That is exactly the bottleneck the field is facing right now. We can map the tissue microanatomy at this unprecedented high-resolution scale, but turning that absolute ocean of spatial data into actual usable clinical knowledge, that's incredibly difficult.
Yeah, I bet.
Traditionally, computational biologists have tried to solve this information overload using two distinct approaches. So the first one is clustering.
Which is essentially just phenotyping, right? You group cells together based solely on what molecules they have inside them, and you just sort of ignore where they actually live in the tissue.
Precisely. The core assumption there is that all the relevant information about a cell is captured entirely by its own internal profile. But that totally ignores the environment.
Right, context matters.
Exactly. A cancer cell acts very differently when it's surrounded by hostile immune cells versus when it's surrounded by, supportive structural cells. So the second traditional approach is neighborhood niche analysis. Tools like Histocat try to figure out who is sitting next to whom in the tissue.
I have to pause you there because neighborhood colloquialization seems inherently flawed to me.
Oh, how so?
Well, it's like assuming two people are best friends just because they happen to be standing next to each other on a crowded subway car. Just because a T cell is physically next to a tumor cell doesn't explain their actual biological relationship.
That is a great analogy.
Huh.
Just being next to each other isn't enough.
Wait, what about graph neural networks? GNNs, they are built specifically for modeling complex networks, right?
They are. And GNNs are incredibly powerful at learning representations from complex neighborhoods. But they have two fatal flaws when it comes to clinical diagnostics. First, they are data hungry.
Meaning they need huge training sets.
Right. They require massive amounts of labeled training data to perform reliably, which we often just don't have for specific rare tumor microenvironments. Second, and honestly more importantly, they are black boxes.
Meaning they give you an answer, but they can't show their work.
Exactly. A GNN might predict that a patient will resist therapy, but it produces these mathematical embeddings that are just not explainable to a human. You can't ask the algorithm why it made its decision.
And in medicine, that's a deal breaker.
Totally. If you can't communicate the biological reasoning to an oncologist, the tool is practically useless. We need explainability.
We need to know the why so we can actually treat the patient. So this brings us to the core of our deep dive today. We are going to look under the hood of a brand new computational method called Kasumi.
Yes, Kasumi.
It is designed to learn tissue representations by identifying persistent local patterns of complex relationships. It doesn't need massive data sets, and crucially, it shows its work. We are going to break down exactly how Kasumi operates, detail by detail, and see how it's predicting cancer patient survival way better than the standard models.
It's a fascinating shift. To really understand Kasumi, we need to start at its foundational architecture. Kasumi actually builds upon an existing mathematical framework called MISTY.
That stands for Multiview Intercellular Spatial Modeling, right?
Yep, that's the one. MISTY is brilliant, but it extracts global relationships, meaning it looks at interactions across the entire tissue sample all at once. Kasumi pivots. It narrows the focus to find highly localized multi-view relationships.
Let's visualize this multi-view concept because it's the engine of the whole system. For you listening, think of a single cell inside a tumor as a house.
Okay.
A house. Kasumi looks at this house through two distinct mathematical lenses, or views. View number one is the intraview. This is intrinsic. What is happening strictly inside the walls of the house?
Right.
If the data uses cell types, this is a simple label like,
Yeah.
This house is a macrophage. Or it could be the exact vector of biomarker proteins currently active inside that specific cell.
And then it layers on the second lens, which is the pair view. This captures the broader tissue context like who lives in the surrounding neighborhood.
So it's looking outside the house.
Exactly. For example, the algorithm might calculate the pair view by summing up the identities of the 10 nearest neighbors. Or, if we're looking at raw protein markers, it calculates a distance-weighted sum of those markers in a specific radius, say 100 pixels.
Let's clarify distance-weighted real quick. That means neighbors who are physically closer to the house mathematically scream louder in the algorithm than neighbors on the edge of the radius, right?
You got it. The closer the neighbor, the heavier its influence on the pair view score. So Kasumi has these two views. To actually scan the entire tissue map, it uses a sliding window mechanism.
How does that work?
Imagine taking a square frame and sliding it inch by inch across the tissue sample. Each window captures a contiguous spatial patch. And critically, the frame slides with a 50% overlap.
Wait, why overlap it so much? Doesn't that just double the computation time?
It does increase computation, yeah, but it's a necessary trade-off. The overlap ensures we don't accidentally drop a crucial biological interaction right down the middle of two separate windows.
Oh, that makes sense. You don't want to cut a conversation in half.
Exactly. Now here is a vital mechanistic detail. If a sliding window happens to land on a sparse area of the tissue, and it contains fewer cells than the number of features the algorithm is looking for, Kasumi simply throws it out.
That makes total sense. You don't want to force a machine learning model to find complex relationships in an empty lot. It prevents the model from overfitting to meaningless background noise. But how does the math actually uncover the relationships within a valid window?
It uses a machine learning ensemble called random forests. Specifically, 100 decision trees working simultaneously.
I always think of random forests like trying to perfect a really complex recipe. You have 100 different chefs testing variations of a soup. The algorithm is basically asking, if I know how much salt is in the broth, the pair of you, how much does my uncertainty about the final taste, the interview, drop?
That is a really great analogy.
It's looking for the feature that reduces the mystery or the variance the most.
That is the perfect way to explain variance reduction. Statistically, the algorithm calculates the reduction in target variance provided by each predictor. If knowing the abundance of a specific molecule in the neighborhood drastically reduces your uncertainty about what's happening inside the target cell, that specific relationship gets a high importance score.
Okay, so it scores everything.
Right. Kasumi then takes all these scores and uses a mathematical blending technique, an L2 regularized late fusion linear meta model, to combine all the predictions.
Okay, let's translate that math into plain English for the listener. It's a blending technique that ensures the inside of the house and the neighborhood are weighed fairly. The L2 regularization acts like a volume knob, right? It prevents one super loud variable from completely drowning out the subtle, nuanced relationships.
Exactly. It keeps things balanced. And that mathematical fusion leads us to the final output of the algorithm. Kasumi organizes this data into 3 specific levels of representation.
I use the mnemonic SCP to remember these 3 levels: signatures, clusters, and persistence. Let's walk through them. Level 1 is signatures, right?
Right. Level 1 is signatures. Every single valid window on the map gets assigned a matrix of importances. This is basically a spreadsheet of predictor-target relationships. And the algorithm is strict. It filters out any relationship that explains less than 1% of the variance.
Okay, so it drops the useless data. Then we move to level 2, which is clusters. Kasumi takes all these windows across all the patient samples and builds a giant web. It links similar windows together using a metric called cosine similarity.
Yes, cosine similarity is key here.
I want to highlight why cosine similarity is used here instead of standard Euclidean distance. In biology, we don't always care about the sheer volume of a protein. We care about the ratio.
Exactly, the relative amounts.
Right, like if one cell has 10 units of protein A and 10 of protein B, and another cell has 100 units of A and 100 of B, their ratio is identical. They're doing the same biological job, just at different scales. Cosine similarity measures the angle between the data vectors, capturing that ratio perfectly.
That is a crucial distinction. It finds windows that are biologically trending in the same direction, regardless of their raw magnitude. It then uses a graph algorithm called Leiden community detection to group those similar windows into tight-knit clusters.
Which brings us to the final level persistence, the ultimate filter.
Right. Kasumi throws out any cluster that doesn't appear in at least 10% of the patient samples, or a minimum of 5 samples.
So if a pattern only shows up in one weird anomaly of a biopsy, Kasumi ignores it. It demands robustness. It only keeps the persistent biological truths. So let's do a quick recap. If I'm tracking with you, the workflow is Kasumi scans the tissue with an overlapping window. In each window, it measures the inside of the cells against the surrounding neighborhood. It scores how important those relationships are, groups the similar windows using cosine similarity, and demands that those patterns exist across multiple patients before it takes them seriously.
That is the Kasumi mechanism in a nutshell.
It makes you wonder, if you're listening to this, when you look at the data in your own field, whatever industry you work in, how much of what you are analyzing is just spatial noise? If you applied a strict persistence filter, what hidden, robust relationships would suddenly become obvious?
Well, to see those hidden relationships in action, we have to look at the clinical evidence. The researchers didn't just build this algorithm in a vacuum. They put Kasumi to the test against several baseline spatial algorithms across 3 highly detailed oncological datasets.
These were real patients with high stakes.
Very high. The first was a DCIS cohort, which is a precursor to breast cancer. This one had 58 patients, 44 non-progressors and 14 progressors. They looked at 17 sublineage labels and 38 markers.
Okay, that's a solid data set.
Then the second was a cutaneous T-cell lymphoma cohort, or CTCL. These were 29 pretreatment patients facing a heavy immunotherapy drug, a PD-1 blockade. 14 responded, 15 didn't. They had 21 labels and 56 markers. The question was, who will actually respond to the drug?
And the third?
The third was a breast cancer cohort testing sensitivity to hormone therapy. 30 patients, an even split of 15 sensitives and 15 resistant, with 27 labels and 33 markers.
And how did Kasumi perform compared to the older models like WCC or CSCA that just look at cell composition or basic colocalization?
Kasumi crushed the baselines in predicting patient outcomes across the board, measured by AUROC. But what's really highly instructive here is how the algorithm handled window size. They discovered a concave relationship.
Meaning there's a strict Goldilocks zone for context.
Exactly. If you set the sliding window too small, you have 0 context. It's just isolated cells again. But if you set the window too big, the localized microenvironment signal, the actual conversation between the cells, gets completely drowned out by the global noise of the entire tissue.
So what were the optimal numbers?
Kasumi found the optimal balance, identifying 33 persistent clusters for the DCIS data, 16 for the lymphoma data, and 9 for the breast cancer data.
Let's dive deep into that lymphoma case, because predicting who will survive immunotherapy is one of the hardest challenges in oncology today. To understand how Kasumi proved its work, we need to introduce a metric called SMR, or signed model reliance.
Right. Signed model reliance is the ultimate algorithmic stress test. Imagine you have a massive spreadsheet of all your cluster data used to predict a patient's survival. To find out how critical one specific cluster is, the algorithm scrambles or permutes the data in just that one column.
It just randomizes it.
Yeah. And if the model's prediction suddenly collapses, it tells us that specific cluster has a high SMR. It is structurally load-bearing for the diagnosis.
So applying that stress test to the lymphoma patients, what was the biological breakthrough?
The model found that for patients who failed to respond to the therapy, its prediction relied heavily on 2 specific spatial patterns, cluster 23 and cluster 16. But for patients who did respond, it relied entirely on cluster 4 and 13.
What was fundamentally different between those clusters?
Cluster 4 showed a strong, positive spatial relationship between the tumor cells and a specific type of anti-tumor immune cell, the M1 macrophages. They were actively interacting in the tissue.
And the other ones?
Clusters 16 and 23. They revealed a distinct avoidance pattern, a stark negative spatial relationship between the tumors and those exact same macrophages.
Hold on, I need to emphasize this for the listener. The baseline models just counted the cells. They would look at a patient and say, good news, you have plenty of immune cells, the immunotherapy should work.
Right, they just looked at the total tally.
But Kasumi realized that even if the immune cells are physically present in the tissue, if the spatial architecture shows they are actively avoiding the tumor cells, the drug will fail. It proved that patients with this low immune activity spatial pattern won't benefit from the treatment.
And that is the profound power of explainable AI in medicine. It doesn't just output a binary give the drug or don't give the drug. It explicitly maps the microanatomical failure point.
Okay, synthesizing this clinical win for a quick recap, Kasumi isn't just winning mathematical benchmarks. By fine-tuning that sliding window to capture local context and then stress testing those patterns with SMR, it identified that spatial avoidance between macrophages and tumors is a perfect predictor of immunotherapy failure.
Exactly.
I mean, if you're a doctor, think about how much more confident you would feel prescribing a grueling treatment knowing you could literally see this avoidance pattern mapped out in the patient's tissue.
Absolutely. Which brings us to the final and perhaps most difficult challenge. Everything we've discussed so far relies on a massive assumption.
Which is what?
That we have clean, perfectly labeled data. We assumed we could point to the map and say, this is a tumor cell and this is a macrophage.
Right, but biological reality is rarely that clean. What if the tissue is just a disaster zone? Cancer radically mutates tissue. What if the cellular structures are so degraded or the lineage markers bleed into each other so much that we literally cannot accurately label the cells to begin with?
That is the current frontier of spatial omics. And this is where Kasumi demonstrates incredible flexibility. It turns out Kasumi doesn't actually need categorical cell labels. It can drop them entirely and look purely at quantitative data raw marker abundances.
It's like instead of relying on a flawed piece of sheet music that tries to label every instrument, you just bypass the labels and listen to the raw audio frequencies of the orchestra itself. You are looking at the raw, unclassified protein levels across the spatial map.
That's a great analogy. And the researchers rigorously tested this. They took baseline spatial algorithms that were specifically designed to handle this kind of raw data algorithms like Banks's Y, CellCharter, and UTAG, and ran them against Kasumi on the exact same data sets.
Using just the raw markers.
Yes, and Kasumi still outperformed them on 2 out of the 3 data sets.
Which proves a massive paradigm shift. The spatial relationships between unclassified markers matter far more than just the spatial composition of those markers. Let's ground this with a specific case study using that breast cancer data set you mentioned earlier. Here we are looking at estrogen and progesterone receptor positive, HER2 negative breast cancer.
Right.
The clinical goal is to predict who will be resistant to hormone therapy. What happened there?
When Kasumi analyzed the raw marker data for these patients, it flagged one specific spatial pattern as the ultimate predictor of hormone resistance. Cluster 37. And because Kasumi shows its work, we can open up the black box of cluster 37 to see the actual biological mechanics driving the resistance.
Okay, so what did the intrinsic view inside the cells show for cluster 37?
Inside the cells, Kasumi found very few strong marker relationships overall. Biologically, this indicates a severe loss of normal regulatory mechanisms, which is exactly what you expect to see in an advanced, highly resistant tumor. However, it did flag a coordinated immune pattern involving markers CD44, CD45, and CD68.
Okay, so the components of the immune system are physically present. But what about the paraview? What was happening outside the cell in the broader neighborhood?
This was the breakthrough. In the paraview, Kasumi detected a massive exclusion pattern. It saw a strong negative spatial relationship between CD45, which represents the immune cells,
Okay.
fibronectin, which is a protein that makes up structural stromal tissue, and GATA3, which is a key marker for the tumor epithelial cells.
Wait, I need to push back here because I know a bit about GATA3. Isn't GATA3 generally considered a good sign? It interacts with estrogen receptors and usually indicates a favorable prognosis and a good response to hormone therapy.
In a vacuum, yes, GATA3 is a favorable marker. But context dictates function. Kasumi discovered that the high intensity of fibronectin wasn't just background noise. It meant a dense physical wall of stroma had formed.
A physical wall.
Yes, this physical wall was blocking the immune cells from infiltrating the tumor. And critically, it was altering the availability of the estrogen receptor. Kasumi independently discovered the spatial architecture of what oncologists call an immune desert.
But how did the rival algorithms miss that? What did an algorithm like Banks's see when it looked at the exact same data?
Banks's Y only saw the composition. It detected the high levels of GATA3 cancer cells and clustered them together, likely predicting a good outcome. But it completely missed the spatial organization. It failed to see the fibronectin wall physically separating the immune cells from the tumor. It missed the actual mechanism of resistance.
That is wild. The rival algorithm essentially looked at a castle, saw the gold inside, and predicted victory, totally missing the massive moat that made the gold inaccessible. But how do we know mathematically that this fibronectin wall isn't just a random artifact? How do we know it's a real biological structure?
By utilizing a statistical measure called spatial autocorrelation, specifically measured by Moran's eye.
Let's define Moran's eye simply. It essentially proves clumpiness.
Clumpiness, yes.
Right. It mathematically verifies that these cluster 37 patterns aren't just randomly scattered across the tissue slide like a handful of pepper thrown on a table. It proves that these specific marker relationships clump together contiguously. It confirms we are looking at true, localized, organized microenvironments acting in concert.
Exactly. It validates that the ecosystem Kasumi found is structurally real.
Okay, another quick recap on this final frontier. Kasumi can bypass flawed human labels entirely. It can read the raw audio frequencies of the proteins, and by analyzing the relationships between those raw markers, it discovered a physical stromal wall blocking immune cells, an immune desert that standard composition algorithms completely missed, and it mathematically proves those structures are real using Moran's eye.
That's the power of it.
It really makes you wonder, when we analyze complex data without mapping the spatial relationships, how many structural walls are we completely blind to?
If we pull back and look at the bigger picture of this entire deep dive, Kasumi proves that human biology operates across totally different scales simultaneously. You have sparse, highly localized events, like the fibronectin wall in cluster 37, happening at the exact same time as global, tissue-wide interactions.
Yeah, it's all connected.
Kasumi successfully bridges the gap between a raw, overwhelming list of spatial measurements and the actual biological mechanisms driving the disease. We are finally evolving from merely phenotyping cells to understanding the ecosystems they construct.
And crucially, it is highly practical. We aren't talking about a theory that requires a supercomputer to run for a month.
Not at all. The architecture is highly scalable and parallelizable. Running Kasumi on categorical cell types takes about 8.9 seconds per sample on a standard laptop.
Wow.
Yeah. For the intensely complex raw marker data, it takes about 30.5 seconds. And it scales up effortlessly to massive transcriptomics datasets too, like the PDA-CSMI dataset, which has 300 fields of view and 700,000 cells. It handles it.
We started this conversation talking about the muddy, chaotic waters of medical diagnostics. We wanted to know how to find clarity when you are staring at a billion data points.
And we found that clarity doesn't come from just zooming in closer on a single isolated cell with a stronger microscope. It comes from stepping back and mapping the relationships.
Exactly. Which brings me to a final thought for you, the listener, to mull over today. We learned that in the battle against cancer, the isolated identity of a cell matters far less than the relationships it forms with its neighbors. A cell isn't defined merely by what's inside it, but by the community it belongs to. If a computational algorithm can map the social network of our cells to accurately predict our survival, what other hidden networks inside us are just waiting to be decoded? Keep looking for the connections, and we'll see you on the next deep dive.